Hello everyone. Good afternoon. My name is Anjali Nema, Assistant Professor of Biotechnology in the Department of Biotechnology, TICS Building, TIT College, Bhopal. Today we are starting a new lecture, which is conducted two chapters. This is the index of today's lecture, which we have to cover in this video. First, in Unit 1. Introduction of mushroom biology, in which we have to cover general introduction of mushroom, nutrition and health benefits of mushroom, nutrition mushroom in Ayurvedic and Indian tribal tradition and economy, role of compost material, swapping method, casing of soil, preparation and the harvesting methods. In second chapter, we include the control environment and post harvesting processing, control environment, identification of contaminants, value addition, food safety compliance, quality standards, packaging, vacuum sealing, and storage transportation. And next, method of increasing storage life and reducing the spoilage. And the reference books which we have to included in the last of the slide. This will say how we have to make this PPT. So the mentioned book are there. In general introduction, we have to start. Mushroom is a freshly fruiting body of fungus, typically produced above the ground of soil. It is a it is a two types of mushroom. First is edible and second is non-edible. Non-edible is generally called as poisonous mushroom. Toadstool is a major species of poisonous mushroom. Mushroom is belongs to the family of Basidiomycetes. It is consist of a part. It is consist of a different parts like cap, stipes, gills, and the major part is mycelium. Let's start the structure of mushroom. मशरूम के स्ट्रक्चर को हम कंप्लीट करते हैं टू टाइप से. इसका एक वन होता है वेजिटेबल पार्ट एंड सेकंड इज फ्रूटिंग बॉडी. Vegetable part is the mycelium. It is grow under the soil, which contain nutrition and support the vegetative body to grow. It's support the fruiting body to grow. It is also called as reproductive structure. Reproductive structure, which we have to easily see by our eyes, and it contains of cap, which is called pilus, gills, lamellae, stipe, stalk, anus, rings, vulva. And this cap is a major part which look like a head, and the stipes which we have to support the mushroom, and anus rings which is present on the stalk, and the vulva which is present on the stipes. Second major part is spores. Spores means it is a seed which we have to use for the major growth of the mushroom. It is very important to select a very good basidia, which we have to when we have to put in the growing in the purpose of growing. They have to give us the good mushroom and a good quality. Let's start the life cycle of mushroom. Life in this part, we have to study how the spore is germinate and how the fruiting body is fine. It includes major steps, and these are spore germination, fusion of the cells, then formation of fruiting body, which is called basidiocarp, and karyogamy, which is also called as a meiosis stage. And the second and last is the continuation of the cycle or spore releasing. In this life cycle, we have to take the good seed, which we have to give a good production of the mushroom, which is very important. Important in this, it is the growth of mushroom is depend upon the major major functions or importance is like pH value, temperature, humidity, wind, water, and the seed which we have to use for the germination. Spore germination. Germination, a good um, seed we have to choose, and a major um, uh, substrate which we have to very important to identify a good favorable substrate which is support the fruiting body for the development, and use it is a basically a nucleus which we have to grow, and the nucleus is dividing with two cells, then the fusion happens and the cells was divided, the two or the different nuclei we have to found the formation, and the second step is formation of fruiting body. When it has divided, then the stalk and cap will be formed when the basidiocarp is generally germinated. We have to see the little of stipe and the pilus formation. Second, next one is meiosis. In this, two nuclei of dicot cell fuse and the form of from diploid nucleus, and the process is called karyogeny. It is very supporting to the projection of newly basidiospore. Spore release in one continuous of the cycle. When the mushroom is completely uh, formed, when the basidiocarp is mature, then they have to release into the environment, and the cycle again uh, uh, begins again, and each spore upon landing of the favorable environment. Next is species selection of mushroom. As I told you, the species selection or the spore material selection is very important for the good production of the mushroom. It is very because we have to choose a variety of mushrooms for the edible purpose, and which is very non in the form of non-poisonous. It mushroom we have to use in a very different types like medicinal values. Tribals also use the mushroom, and the types of mushroom are also there. The types of mushrooms is oyster mushroom, button mushroom, sea-tac mushroom, and the gonoderma mushroom. Oyster mushroom is basically used for very much used for the tribes purpose. They are used as a food. It is structurally it is a fan shaped, and the growth required an optimum temperature which is 20 to 30 degrees Celsius, and the high humidity. Uses we have to use in a variety of humans are used to take fulfill of nutritional values like vitamin D, which and vitamin B and D, and the high mineral and fiber content of mushroom. It's very important for the digestion. Second was is button mushroom. This is very commonly used mushroom. The structure is like a button, that's why we have to call it a button shape. It is um, and the growth of this mushroom depends upon the multiple things like temperature. For the growth temperature is 25 to 22 to 25 degrees Celsius, and the mycelium required 16 to 80 degrees Celsius for the fruiting body development. Use the uses of this is it is a very good and rich source of protein, potassium, phosphorus, silicium, and vitamin. It is commonly used as a soup, pizza, and salads purpose. Sea mushroom. This is the native mushroom, East Asia, and the widely grown in the hardwood logs. Structure is the toughy, fleshy, and with a strong earthy aroma. Its growth preferred to 18 to 25 degrees Celsius. It is also called as anti-cancerous properties. Gonoderma mushroom, same as the uh, nutrition value is same as like other mushrooms because they all are non-poisonous mushrooms. Identification and cultivation of the mushroom. When the fruiting body and everything was be developed in this, then we have to cultivate the mushrooms, and the morphologically when we have to see and morphologically cap gills, stipes, pods, habitat, always is also all are completed. Then we have to choose a full sanitized way and a hygienic way to develop to cultivate the mushrooms. Steps of cultivation is just a substrate preparation, swap preparation, incubation, inoculation, fruiting body, and harvesting. As we have to discuss in the life cycle of the mushroom, the whole part of my life cycle is covered in this part. Like substrate selection, we have to select a good substrate for the growth and the humidity temperature for the development of fruiting body as required. Advantages of cultivation is this: it is a quality and food safety, consistent the quality and yearly around the when the mushroom is growth is rich in protein and the properties is good, then we have to take the variety of mushroom whole the year. The cost is very supporting for the produce. Second one is poisonous and non-poisonous mushroom. Which mushroom is edible? They are called non-poisonous mushroom, and which are and the other one is non-edible, which is called poisonous mushroom. The examples of non-poisonous mushrooms are oyster mushroom, sea mushrooms, and button mushroom. Non-poisonous mushrooms are toxic for humans, and the chemical properties compounds such as antitoxin, belatoxin, and other is not support the human body and also cause the abdominal pain, diarrhea, liver failure, and the death also. Common examples of poisonous mushroom is anemiotic or a dead cat mushroom. Next, we have to discuss the nutritional and health benefit of mushroom. As you know, the mushrooms are very rich in vitamins, minerals, fats, health benefits. It is supports human body for a healthy benefits. Mushroom in Ayurveda or Indian tribal tradition, mushroom have a very good source of nutrition. So the and very easy to develop in the tribal areas. In Ayurveda, we have to use mushroom as a medicine for mushroom, and tribal people use as a they are also celebrate the mushroom as a cultural role. Widely distribution economic importance is very important for this because tribal peoples grow more mushrooms and grow sales the in the market, and it is a very good source for their livelihood and security. Role of compost material is this the material which we have to use for the growth of mushrooms. Chemical properties, physical properties, biological properties, and the environmental. Chemical properties, we have to use the chemical which have to support the growth of mushroom. Physical properties like temperature, pH, humidity. These are the physical property which we have to provide for a good mushroom. Biological properties, we have to say inoculation, support the diverse microbial communities, bacteria, fungi, antibiotics. These are also helpful. Environmentally, the compost material and the uh, fertilizers we have to use for a good purpose and the make it pollution free and food free. Economically or socially, the compost material are suitable for agriculture, circular economy models. Farmers are convert crop residues and manure and waste into a value addition inputs, lowering and the cost of the improving soil health longer terms. Process is just like a we have to select a good raw material like a, uh, uh, wheat grass. These are very wheat substrate which are very good for hydrating properties and the other properties. Then we have to mix it for a chopped it for, because very important to chopping of mushroom as the aeration proper, as we have to give the aeration property for moisture purpose. We have to immerse the 60 to 70% of moisture, because mushroom has a hydration properties. It is a very good source of hydration. Screening and storage after the once the compost is mature, it is screed through the sieves and remove the large decomposed particles, then the fine dark brown and early smelling compost. It is stored in a dry and cool place to prevent the recontamination and the moisture loss. Swapping method. The swapping of the seeds material substrate and colonize of fungus like mycelium growth, it is very important, and the swapping tradition and the substrate preparation as the same as we have to discuss in previously, like that. Advantages is grain swap and the fast colonization, flexible mixing, having contamination of heavy contamination risk if not sterile. Sterilization is very important for preventing the contamination. Sawdust is a very good source for substrate and for wood species. And the other one, casing of soil preparation and harvesting methods. The layer role is to maintain the moisture surface and develop the penis. These are the in this PPT. These are the methods of curing the material and preparing the material. The preparation steps are also maintained how the soil is to be prepared. Next, we have to start the unit 2. And next we have to start the unit 2. In this unit 2, we have to cover the control environment and post harvesting system. First unit we have to see whole the process how to mushroom will be germinate and growth. The control environment identification means the controlling environment refers to the regulation of key physical parameters such as temperature, humidity, ventilation, light, carbon dioxide level during various stages of mushroom growth. As you know, I was told you previously, the very important for the mushroom growth is light, temperature, ventilation, and sanitation. Because if we are not providing in the accurate or a moderate quantity, the growth of mushroom is not being good as we want. So it's very important to give the good light. Sanitation is the very important. Why? Because we have to maintain it or grow it without the contamination. The contamination is the whole disturbing process of the mushroom production. We have to provide proper ventilation is the air exchange of C2 and O2 and the level. Second, light, it is very important. Around 300 to 500 lakhs is very important. Luminous sanitation, as we have to discuss the clean surface around, we have to work the use of the clean equipments, clothes, gloves, etc. Identification of contaminant. If our mushroom is going to be face the some contaminants, we have to analyze and remove the contaminant parts. Food fungal contaminants, as we have to grow the mushroom, we have to see the patches around the mushroom like green patches, so we have to uh, prick them and remove the and provide a proper nutrition to the mushroom, so the other part, other mushrooms are not get contaminated by the reference of others. Bacterial contamination, it is majorly occurs in the substrate part because of the moistures. If we have to give the over moisture, it will be slimy or smelly. Discoloration is also seen in this part. Prevention and control. Prevention and control, sanitization and pasteurization of the substrate is very important. Clean surfaces, putting using the uses of aseptic techniques and disinfection. It is the major prevention and control of the mushroom cultivation. Value addition, when we have to grow the mushroom, it is very important to add the requirement which support the nutrition. Requirement of nutrition which support the mushroom growth. It's very important. Dehydration, drying of mushroom in this dehydration, the most preservation method for the mushroom. If you have, we have to take mushroom of whole the year, we have to consume or uh, prevent them. Variety, sun dry or economic freezing of mushroom. These are the major beneficial type of mushroom variety to be have to stored it. Mushroom powder production. It is very important to add the flavors in our food. So we have to dry the mushroom and make it a powder and sprinkles. So it's make a um, powder form and popularity type or used as a popularity types. We have to use these kind of mushrooms as we making a powder like button mushroom, oyster mushroom, and sea mushroom. Mushroom based snacks. We have to use mushroom and a snack variety like chips, pickles, biscuits, and other snacks. Mushroom leathers we have to also use in a leather forms. Mushroom uh, based meat products. We have to we have to use just soya beans, soya bean buddies. So soya chunks. So as we use soya chunks as the replacement of meat, so we have to also use mushroom based product which is likely to be meat but not meat. So it's very good form to replacement meat products with the mushrooms. Food safety compliances. It's very important to add nutrition value or a safety value to good yield of the mushrooms. Food safety purpose value addition like dehydration, good handling of the equipments, follow the GMP rules. GMP is a good manufacture process practices and regulated. Record and audits is also very important to maintain a raw material processing steps clean. By the step by steps processing or safety purpose, we have to maintain the records and keep auditing them. We have, keep certification updates like FSSAI and ISO approvals is very important and applicable for the Indian brands. Quality standard packaging, storage, and transportation. In quality standard, we have to maintain the qualities and quality of which include the physical, chemical appearance, texture of mushrooms, microbial activity or standards, chemical safety, and grading. Like physical properties, like good handling of mushrooms so that they can't be flashy or discoloring, brushing like that. Color is very important to see the mushroom. If we have to do on see this, it's become a spots on their cap. So we have to maintain that same as a texture. If we don't see that, it's becoming the sour or slimy. Packaging is very important for a fresh mushroom. We have to use a plastic tray, uh, free ventilation. Dried mushroom packaging is a illuminated airtight containers, and the labeling includes very much because we have to, it is undercover the FSSAI. rules and information. Otherwise, the licensing member will be punished if we don't have to follow the vacuum sealing. Vacuum sealing is just a reduction of air in the presence of material. Transportation maintain a proper environment or a safety quality to preservation and transportation. Use refrigerator to maintain the low temperature and high temperature humidity. Package mushroom should to be secured and avoid the crushing during the transportation. Frozen and the dried mushroom should be and include include the maintenance purpose and the moisture fluctuation. Method of increasing storage life and reducing spoilage. Harvesting right can cleaning storage, temperature control humidity, packaging techniques, application of preservation and the freezing time. All are we discussed in the previously. Like cold storage is very good for the growth of mushroom. We have to maintain 1 to 4 degrees Celsius and the humidity at 80 to 95 for the prevention of the microbial growth. Control humidity. It is very important to ventilation remove the excess of CO2, which gives the which is very important because of excess of CO2 it will be spoil the whole production. Packaging techniques, we have to use the vacuum packaging um, to remove the air and it will be slow the contamination. Application purpose, application preservation, food safety is very important for the edible purposes, edible and the freezing drying. The dehydration and the clean all the tools storage is very important for the good transportation. Like workers should be wear gloves, masks during the processing and regulate the monitoring for microbial load in the storage areas, prevent the outbreaks of spoilage. Rapid post harvesting handlings minimize the time between the harvest and cooling, rapid cooling, slow respiration and microbial growth. Avoid stocking and the comprehensions of mushroom during the storage and transport to the prevent brushing and the accelerated spoilage. Lastly, these are the references which I have to use for making my PPT. Thank you so much for this lecture and listening me a concentration. Concentrate. Thank you.